I'm Wendy Hartsock, science and peptide enthusiast. In this episode of Exploration Science, I sat down with Professors Anna Maria Papini and Paolo Rovero to discuss their research interests around peptides in immunology, diagnostics, and cosmeceuticals. They also talk about their work to improve access to industrial scale production of peptides. Now, how long have you both actually been working together?
Oh, it's a very, very difficult question. We were together in the high school.
Oh, wow.
So we were together in the high school, then the university, but then we had two different pathways till 203. But Paolo can start and saying what is the pathway, but that's a story. So it's a long story.
Okay.
And by the way, we are married. No, okay. We can say the real common work started in 2003 when I came back to Florence professional. At the beginning of my career, I've been in a pharmaceutical company, still working in peptide here in Florence, but it was not a university, it was the company. Then in 1991, I left the company and I moved to the National Research Council in Pisa, Tuscany, but on the coast, still dealing with peptides, but peptides in immunology. Before in the company was pharmacologically active peptides, neurotransmitter antagonists, this kind of pharmaceutically relevant peptides. Then again, in 1998, I was appointed the professor of medicinal chemistry, and I moved to Salerno, which is south of Naples, in the south of Italy. We're still working in research in the field of peptides and already starting a collaboration with Anna Maria, but I was mostly teaching, I must say. Finally, in 2003, I get the position here in Florence as a professor of medicinal chemistry at the School of Pharmacy. At that point, Anna Maria was already in Florence in the Department of Chemistry. We started an interdepartmental laboratory because formally we are in two different departments and two different schools. I'm in the medical school as a pharmacist. She's in the School of Science, Chemistry. And from that moment, the interdepartmental laboratory of peptide and protein chemistry and biology went on as a single research unit. And since that period, we are really collaborating much more strictly.
And from my side, I started peptides from nothing at the University of Florence with my PhD. And this is why I did half of my PhD with an international program, one period in Quebec, in peptides and immunology, and one period at the Max Planck Institute for Biochemie, for biochemistry in Munich, under the supervision of Luis Moroda. And once I came back to Italy in 1990, I started a laboratory of synthetic peptide synthesis, in the Faculty of Medicine, because I was associated to a professor of organic chemistry for MDs. But then I moved to the scientific pole that has just been created in 2001, where I launched the Laboratory of Peptide Chemistry. But in 2009, I launched another Laboratory of Peptide Chemistry in Région de France, in CY Cergy Paris University, where I have been half-time together with the position here in Florence, to launch peptides there, and then joining the two labs. But since 2020, I am based essentially here. not only because of COVID, but because I did my job there, and then I had to push once again here at the University of Florence, working in the interdepartmental research unit of peptide and protein chemistry and biology with Paolo.
Nice. And what are sort of the overarching goals of your research?
So from my side, as I have been always in the chemistry department, I can say that I have been always focused in having all the possible synthetic strategies in hands to produce peptides, but particularly modified peptides with sugars, with lipids, with exotic conformational constraints, thanks to also particular organic chemistry strategies like staple peptides, therefore using click reaction on side chain to side chain cyclization, always with the idea in mind that the organic peptide chemist should produce precise molecules for different applications. And one of the main subjects I have been always involved, also thanks to my education during my PhD, was to work on chemical immunology, so characterization of antibodies and T cells by using peptides, possibly in investigating the mechanism of immunomediated diseases. And Paolo, probably you can more?
Indeed, even if I've been built as a synthetic chemist, as I mentioned before, I started my career in a pharmaceutical company, so I was much more focused on applications of peptides at the beginning as therapeutics. Very early, I must say, because it was the late 80s in the last century. So in 1986, 87, we started to be interested in tachykinin antagonists as potential drug. At the time, we were competing with group in Merck, in other leading pharma companies. Then 5 years later, I left the company and I moved, as I mentioned, to the National Research Council. I also started to be interested in peptide immunology and the applications. In that case, I moved toward diagnostics. So peptide as active ingredients for in vitro diagnostics test, particularly for the detection of antibodies, both antibodies as biomarkers in autoimmune diseases, and this is exactly the field in which our collaboration became stronger. And more recently, anti-drug antibodies for those patients which are treated with therapeutics antibodies. And always the peptide as the active ingredients, let's say the probe which enabled the characterization of the antibodies. Even more recently, in the last almost 10 years, I say I downgraded. At the beginning, I downgraded from pharma to diagnostic, and more recently, I further downgraded from diagnostic to cosmetics. Peptides as active ingredients of cosmetic products, particularly in the field of collagen turnover modulators, so anti-aging product. I say this is a downgrading, but I do believe that we should be realistic. A relatively small academic group cannot compete in the pharma arena. It's much more realistic to operate in the diagnostic field. And in fact, we started a company in that field and we bring a product on the market. And it is even simpler somehow, and the time to market and then the effort needed to operate in the cosmetic arena. Even if we do not forget our, let's say, tradition, we pay a lot of attention of the quality of peptides, the characterization of the integrity, the purity, the characteristic of the peptide, how it has been synthesized, and particularly referring to the field of cosmetic, the scientifically sound proof of the claimed action. We can use the term cosmeceutical. It's diffused now, this term, cosmeceutical product, even if this term, this definition is not recognized by the regulatory authorities because it's too much on the border, let's say, between pharma and pure cosmetic. We interpret, we use this word, this definition, because we try to use a pharmaceutical approach to the development of active ingredients for cosmetic use. So if we claim that our peptide can be modulator of collagen turnover, inhibitor of collagen degradation, we do have performed in vitro assay to show this activity. We are absolutely certain of the chemical and of the chemical characterization of these peptides, while sometimes in the field of cosmetics the word peptide is used because it's fancy, referring to protein hydrolysate or some stuff which is not exactly
right. You know, I used to work for Allergan. I was a medical science liaison in the aesthetics division, and so I supported Botox and Juvederm and Kybella and Cool Tone and Cool Sculpting. But yeah, it is interesting to your point that, you know, I would go to like different offices and I would look at the like elixirs and serums that they had to treat patients, and it'd say like, you know, peptide, and then I would look at the ingredients and I'd be like, there's not a single peptide in here. There's just some amino acids. Or it would be like peptide 3, peptide 4, like, you know, it was like, so yeah, kind of nebulous and like, what is this?
Absolutely.
So oh, go ahead, Anna Maria.
No, please.
Oh, okay. I was just curious for the cosmeceutical peptides. Are you looking at injectables or topicals, or what is the sort of the area that you are interested in?
We are working on topical peptides, because with the collagen turnover modulator, the target is skin aging, including photoaging or pollution, the aging due to pollution, which can be treated topically with creams. Which is easier. You have less, let's say, interference or risks of systemic effects. So I feel much more comfortable dealing with the peptide for topical use. This could be associated to more invasive, let's say, surgical interventions like the suspension thread or injection of filler. In that case, this kind of cream or topical products could be suggested as post-surgical treatments to sustain collagen regeneration and formation. Yeah.
Oh, it's very fascinating. It is funny though, because on the surface we think of like aesthetics as like not science, and then once you get into it, even the publications that like the number of publications around this area are vast, and the science is very robust. So yeah, that's a very exciting area. So then, total different segue here. How did you get interested in manufacturing scale production of peptides?
So essentially, always with the idea that that peptide science requires good organic peptide chemist, and this is not only me that says that, but we have to go back to Robert Bruce Merrifield, who has always claimed that it is not because of automatization that we have to think that we can overcome the chemistry that is behind each peptide molecule. That is a little bit what, in a certain way, particularly in Europe, but originally also in US, the chemical societies believe that this science could be in the hands of anybody. Okay, I'm not saying that everybody can enter in the field, but you have to know the chemistry that is behind that. And therefore, it's very important that once you decide to work particularly in the academy, in the field of peptides, you have a duty to let know as much as possible to the new generation the chemistry of peptides, how peptides can be produced, and going from the small scale to the large scale. Why? Because once you are working in the small scale, you can, in a certain way, not looking at those possible side reactions that can occur, but you have to know them. Because once you shift to the large scale, you have to limit these side products that sometimes are difficult to be eliminated from the stuff. Yeah. So therefore, essentially, it's absolutely important, even if you are working on the small scale, to have the best chemistry. And therefore, working with efficient strategies, both in terms of protection strategies, on the type of strategy you are using. So, for example, rapid, higher temperature compared to working at room temperature, or also changing the coupling reagents, also having clear control of the starting materials that can affect your synthesis, particularly once you are working in solid phase. It's a challenge, and you have a duty to transfer this technology not only to those that are working in R&D, but mainly also those that are in the production. Because sometimes you can have small and medium enterprises that decide to have a niche of market that is around one or two single peptides that can make their market interesting, but they probably are not good peptide chemists because they never manage these molecules. So therefore, as people working in the academia, we have not only the duty to build young generation, but also to help those companies that want to enter in the field of a peptide used in different applications as an active ingredient. So therefore, this is the reason why we have always associated our applied projects to a wonderful chemistry, the best one to be performed, to have to be sure that this material is the real one at the best yield, with the best yield, and with the limiting the possible side products that can affect the final activity of the active ingredient.
If I can say, well, there is another aspect that pushed us toward, let's say, process chemistry of peptide. And this is linked again to our interest toward diagnostics and cosmetics. And it's what we feel, at least here in our, let's say in Italy certainly, but probably also in Europe somehow, a gap in the possibility to obtain the production of peptides, the custom production of peptides, in the scale of the 100 of grams up to 1 kilogram, and which is typically 100 of grams or tens to hundreds of grams, the scale needed for these diagnostic companies or cosmetic companies which use small quantities of the active ingredient. And so this was one of the aspects while as researchers we were proposing new peptides to be used both in diagnostics and in cosmetics to also help in obtaining the quantity which are needed. And this brings us also to approach the problem of GMP production, which is a huge problem, typical of the pharma industry, of the preparation of active pharmaceutical ingredient, but which are becoming more and more important, certainly in cosmetics, particularly if we look at the regulation, which are apparently more stringent in certain Asia-Pacific region countries. And also in the cosmetic field, they are, in the diagnostics field, they are moving toward more stringent rules. Anna Maria.
If I can add something, very often also those researchers involved in R&D activities require sometimes peptides for clinical phases. And big companies usually producing active chemical ingredients have too higher cost for these phases that are in a certain way the same cost that later on could be applied to those that they have to produce the real active pharmaceutical ingredient for the drug, for the final drug. And therefore, companies involved in these R&D phases with clinical phases cannot afford this cost. So therefore, the idea of the University of Florence is to help those companies involved in the clinical phases in hosting directly in our buildings in the context of what we define joint labs, means that the dedicated labs like the one we have in our University of Florence, the name is Models. Essentially, we have researchers working together with our academic researchers dedicated to help them in producing those quantities that will be used for the clinical phases studies.
Can you talk a little bit about that process of, and I'd like to talk about active pharmaceutical ingredients and then move over to the cosmeceuticals and the diagnostics, but just to start with the process of going from, making a peptide at the small scale to the large scale, and also making sure that it's GMP compliant.
So, Paolo.
No, go ahead.
So, essentially, the ideal situation is that you have an instrument that once you are working to the 5 millimole scale, you can go to the at least 70 millimole scale, maintaining the same reproducible purity. Because, and if you have this possibility, you have done bingo. Why? Because you can set up the procedure simply in the small scale without increasing so much the cost. Because once you are working to the 5 millimole scale, you can do several mistakes. I say mistakes in terms of not taking into account those side reactions that have to be avoided. Once you do not have reproducibility from the 5 millimole to the 70 millimole scale, the side products once formed in the 70 millimole scale, if they were not abolished in the 5 millimole scale, will affect drastically your income because of the high cost of a large quantity of material you were producing. So therefore, having ideally instruments that can help you before going to the GMP facility to produce 5 up to 70 millimole scale, you can essentially have the description on how, once the system has been put in GMP, they can directly go into the market with that product. So therefore, and having all the processing essentially inside the same instrument is also the ideal situation, both in terms of limiting also the operator mistake and in not contaminating the product because of the interference of the environment. So therefore, managing as much as possible inside one instrument that has been put in GMP, but that it's the same instrument that you can have in a pilot lab, not in GMP, is the ideal situation.
Okay.
I would like to add in this setting not only the GMP plant, the pilot plant, but also the research laboratory. One idea that we have had for many times, not easy to implement, but is that if you can have quality as an important parameter, even in the research activity, it will be quicker to move forward the production of the pilot plant in quality. And we try, it's not easy at all, but we try to have our student, our laboratory in the research phase, working in quality and with a mentality that is ready to move toward a GMP or a higher level of quality. And a kind of setting or a kind of instruments that can facilitate this transition, help in setting, putting in practice this idea.
Yeah, I still have a degree in medicinal chemistry as well, and I remember learning like, okay, well, you just you find the molecule that works, and the process chemist will fix the chemistry. And I like your approach is no, just do it right the first time in the research. Yeah.
Yes, it's exactly that, and this is why we call this idea of joint lab in quality PET-Farm. So, and models is essentially the word that reminds that this is a model of laboratory that even if it's working in a separate lab, but always in the academic environment, can have all the facility to operate in quality and then to transfer to the plant that is working really in GMP. But
right.
Same type of instrument.
Yeah. Can you also just sort of give an overview of what it means to be GMP, or like kind of what it takes to get GMP status?
Not easy to simplify, but okay, GMP are good practices, good manufacturing practices, and there are also GLP, the good laboratory practices. This means that you have some, let's say, guideline, stringent, more or less stringent if you start from a GLP or from our concept to work in quality in the research lab. But the idea is that you have some guidelines, then that you should follow this guideline to avoid errors, to avoid to design the quality of your product from the beginning, rather than arriving with the final product. Okay, there is a deletion, there is an impurity. Let's go back. Okay, no, let's try to preview, to analyze at the beginning. This is the general concept of working in quality. And as we were saying, should be done from the very beginning as a principle, but also if you start from the research activity and you try to give to your students and to your people in the lab the mentality to think before starting to move the ends in the lab.
Yeah, absolutely. So I was reading. Yeah, go ahead.
Sorry. And this is what is also very important, is that working in quality means also to limit those side products that can be formed and that are absolutely sequence dependent.
Right.
Dependent means that in any case you can have a process that is imagined to be done because you can envisage what are the future side products that can be formed and limiting them. But in any case, once you have this chemistry, you have always in hands the possibility to reproduce these side products in order to check them and to be sure or to guarantee that these are above, that are downgraded and are not affecting the final purity required by the GMP process.
Well, and you said two, I think, keywords, like, so control and optimization. And so I was reading through the publications on, it's always so hard for me to say it, Eptifibatide. Is that the right way to say that, peptide? It's a cyclic 7 residue peptide. And so there's one publication really talking about going from the 5 millimole scale to the 70 millimole scale. And then there's another publication, and I'll link both of those, by the way, to this podcast so folks can pull up the links. And the other one's really about making the cyclization process the most efficient as possible. And what I enjoyed going through those publications was really getting a feel for what it takes to make something GMP. So like to your point, like you have to make sure that the solvents aren't going to have any impurities and cause, even if you're precipitating into ether, what ether do you use that's going to be compliant and give you the least impurities? I mean, it's so much more than that. I mean, so those publications are really, really good resources for anyone who wants to know what does it take to get to a GMP process. And yeah.
It's a proof of concept.
Mm-hmm.
Go ahead.
It's a proof of concept that you have to follow a precise protocols to limit all the possible drawbacks of this synthesis. And I say the devil is always ready to come and affect your final purity. And the devil can be hidden in a solvent, as you say, in a starting material that can be the building blocks like the protected amino acids, coupling reagents, but can be also storage of all the reagents, because the same storage can, in a certain way, affect the final grade of purity required to obtain the final product.
Yeah. So look, thinking about the active pharmaceutical ingredient versus the cosmeceuticals and the diagnostics is, what is the difference between the stringency on compliance?
There are important differences, and the three products, the three kind of products are in the order that you mentioned. So the top is the active pharmaceutical ingredients. The cosmetic or cosmeceutical peptides are somehow approaching progressively to the pharmaceutical ingredients, because there are rules becoming more stringent, certainly here in Europe, but also in the States and Canada, and as I mentioned, in the Asian countries. The difference could be linked, in the case of cosmetic, to the fact that if the ingredients are below a low percentage as component of the final cosmetic product, the stringency is less important if these components are below the 3% of the total composition of the cream. For the in vitro diagnostics, there is even less stringency, particularly for the field in which with which we are more familiar, the use of peptides as probe for immunoenzymatic assay, or the kind of assays used, in vitro assays used to measure and to characterize antibodies. But this is true in all the three examples. rules are becoming more stringent, and even the regulatory authorities, the pharmaceutical regulatory authorities are moving toward more stringent requirement in terms, for instance, of characterization of impurities. In the case of peptide therapeutics, they have their own set of rules that are, including the FDA, the authorities are revising and updating these. So they keep up to date the preparation methods and the GMP rules will be more and more important, including the analytical aspects. As I mentioned, characterization of impurities also at the very low quantity of impurity. So there is a lot of work to do. And once again, the attitude to say, okay, someone else will take care of this later, in our opinion, is not correct. It would be much better to have in mind this kind of problem also at the research level, and thinking about students or people that we are building in our lab. If they already knows, if they are already aware of this kind of problem, it will be easier on one end for them to find a job later, for instance, which is not, which is important.
Negligible.
And also to have better products and to obtain, since we are interested in the application of peptide, to obtain from research peptides more easily amenable to become products or component of products.
What do you think as far as technologies that will be most beneficial to this process? What are the coming technologies that you see to sort of help in all of this?
Technology is fundamental, both in synthesis and in the analytical field. In synthesis, all those systems that enables to have a synthesizer, automatic synthesizer with a stringent control, even moving toward larger scale. So, for instance, eating. If you eat, the control of the homogeneity of the eating, again, not only in the small laboratory scale reactor, but also in larger reactor, homogeneity of the eating. The ramp of the eating, the control of how to stop the eating at the required level. These are all important aspects. The precision of the fluidic, which move not only the solvent, but most importantly the active, the activators, the protection steps, etc.
Washings.
The washing
Washings, because once we work in solid phase, a very important phase to obtain good material is that washing is efficient enough, because you have to know that once you do not wash the side products that can remain inside the beads, can affect the final purity of the product once the product is clipped from the resin. Mm-hmm. So therefore, the efficiency of the synthesizer is an extremely important part, takes an important part. Probably you can say something about the chromatographic techniques that can be used.
As I mentioned, as it is required to characterize impurities at very low concentration, the efficiency of separation techniques become fundamental. The possibility to have complementary orthogonal chromatographic techniques enabling to look for hidden impurity which can in the standard system in reverse phase HPLC can coelute with the main product, or different impurities that can coelute each other are indeed the problem. In this, I think that I'm not particularly expert of chromatography, but this is another key issue for the quality, to demonstrate and to check for the quality of active peptides.
Right. So will the model lab that has the large-scale production of peptides partner with a chromatography unit, or are you bringing the chromatography into your space?
We have instrument for chromatographic purification inside our building, because it is absolutely important to have these techniques since the very beginning of a process.
Mm-hmm.
Once you are synthesizing, you have to check the possibility of formation of some side products. And therefore, the process has to be controlled continuously. Therefore, even if you can, how can I say, think that you are wasting time because you have to control on some specific couplings that can be difficult. On the contrary, controlling and therefore eventually changing the chemistry during the synthesis can help you later on to develop the right synthetic strategies to be applied for the larger scale. So chromatographic techniques since the very beginning can help in also deciding how to modify your chemistry.
Okay. What were some of the sort of like surprises that came out when you started scaling up from the research scale 5 millimole up to 70 millimole? Was there something that happened, you came across and you're like, oh, that was unexpected? Or was it pretty much like, nope, we expected all of this?
For me, it was unexpected. With a larger synthesizer in an automatic system I used was the reproducibility of a system in terms of purity of a crude. I can say that it was unexpected to have such a higher purity in large scale. I have been working with large scale in liquid phase, because once I was during my PhD in Munich, essentially I learned what are problems once you are working in production of 10-gram peptide and going to 100-gram peptides. Sometimes working in liquid phase leads you to decide what is the maximum of a scale you can work, because in liquid phase everything can be slower and therefore can lead, once you increase the scale, to have a higher increase in side products. Once you are working in solid phase, if you control, on the contrary, the side products, the chemistry is dead, and you can go up to this 70 millimole scale, also increasing the purity. Probably the increase in purity of a crude comes from the increase in washing that we experienced with a large-scale synthesizer. But this is once again the difference between working in solid phase and working in liquid phase. Yeah, please.
I would say the let's say reproducibility and of the instrument become an important point, because if you can work and perform all your synthesis in the machine, should be, if the machine is robust enough, much more reproducible as compared to a liquid phase synthesis, which I feel it's much more operator dependent. Even if you have your GMP and your protocols and your SOP, it should not in principle be operator dependent. But I would feel, and our experience confirms this feeling, that the machine helps, the technology helps in this respect.
I mean, anytime there's human involvement, there's variability, like you said, even if it's a process where you have it, step 1, step 2, step 3, but yeah, everyone can do it slightly differently. So.
Yes.
So do you intend, so you'll have this laboratory where companies can come to you and say, we need production for our phase 1 study, but then you've also talked about getting sort of protocols in place. So is your goal to have like a central repository of protocols for companies to use for production of peptides? Or I guess I'm wondering how you'll interface with different companies as they start making more peptide therapeutics.
So we have repository where that we share in NDA, in which essentially we report all the different steps, and once the steps is set up, we define as the final one to be used. And then once this has been set up in our laboratories, it is transferred to their plant in order to verify and definitively go to the next step. This is how it works. It's evident that then we acquire inside the lab a big experience, that it's going further to the possibility of preparing peptide A or peptide B or peptide C. But this is typical, and this comes also from the philosophy of our PET lab, because since we started our PET lab in Florence, we launched a small spin-off. The name is SPPS, that was launched in 2003, where we performed custom synthesis for some special customers. What are special customers? Customers that are more interested in those peptides that they believe that can be helpful, but then we help also to design that peptide to achieve their goal. This means that in all these years, we acquired a large experience in synthesizing and preparing and solving so many different problems. And this is what, in my opinion, it's extremely exciting, because it's a know-how that it's now in the hands of this lab. This means that also with a lot of colleagues coming from different countries, working in the academy of different countries in Europe, they decide to send their student to prepare a specific product they are interested in having for their research purpose. So this means that having, in my opinion, such a facility and possibly a network of facilities like the one we have could help a lot to push peptides in many different subjects, because otherwise we can have the problem that a new researcher, a young researcher who decides to enter for the first time in peptides has to reinvent peptide chemistry. This is a little bit the problem that sometimes is around having more people in academia working with peptides. So having networks of peptide facility like our pet lab could help in let the young researcher joining the different ones and arriving faster to the final products they are interested in. And this could later on push also companies, new companies, in entering in the field.
And, so along those lines, so for the publications, again, that I'll link to this podcast, where you've really optimized the synthesis of Eptifibatide, not only the solid-phase synthesis, but doing on-resin cyclization where it's fully automated. What do you think are the sort of possibilities, but then also limitations of the complexity and length of peptides that we can manufacture with this instrumentation?
You can think about also very exotic. May I speak about click chemistry?
Yeah.
Triazolyl-based cyclopeptides, thanks to a click chemistry that apparently people can imagine that it's more in the hands of big industries trying to enter to produce this type of thing. I am not speaking about big pharma, okay? I'm speaking about small companies that can think that a stable peptide can solve a biological problem, stabilizing the final peptide that could have ideally an interesting bioactivity. So efficient synthetic strategies can be applied also to these exotic type of peptides. So if I can use a term that, in my opinion, sometimes organic chemists go a little bit too far away, a peptidomimetics can be simply that peptide that has the majority of the sequence in the native sequence, but then a small part that is mimetic, like the triazolyl bridge, that is a mimetic of a lactam. Therefore, the constraint can mimic a beta turn or an alpha helical structure that are relevant for the biological activity.
And again, it should be underlined that the solid phase methods is not a limitation. We do perform also this kind of cyclization in solid phase. We have not faced for the moment the large scale in solid phase, but still the idea to exploit and to take advantage of the solid phase also for this kind of more exotic chemistries is an important possibility and important opportunity.
Well, is there anything that you wanted to talk about, maybe that I didn't touch on or ask about?
I do believe that we have to convince the community, the scientific community, particularly the community that can be involved in those funding agencies, in organizing at the best networks of peptide chemists, to organize at the best the possibility to host young researchers, to push this type of synthetic chemistry with a large control of the products that are produced in order to push this fascinating world that is the world of peptides. Because peptides are natural molecules and have a large variety of functional groups, most of the functional groups present in organic chemistry, and solid phase synthesis, the genius of Robert Bruce Merrifield, is now what he defined in his papers after the after what he published in 1963, in 1969 was already saying that a gold mine was awaiting organic chemistry. And that's true. His genial idea is to have very nice products, and all the possible industrial producer of building blocks, reagents, solvents, and so on, are contributing technologies of instruments, are contributing to help in producing peptides that have 1,000 possibilities of applying in most of the interesting in the interesting field where these molecules can be used.
And this is a message also, and most importantly, for young people, for young people, for students interested in chemistry. Look at peptide, look at proteins and their application in life sciences, but also in industry. We mentioned pharmaceutical, cosmetic, and
materials.
Okay, I was adding, there are many other fields in which there are openings, incredible openings for peptides, and we need, once again, a strong chemistry on the back of this.
Absolutely. And if I can also say something, if personally I was able in now we can say 30 years from nothing, starting and being able in an academia in Italy, where our country does not invest so much as other countries do in research, I can say that everybody can succeed. I am not a superhuman, and so therefore I am a normal human, and therefore this is only to say if we want, we can.
Absolutely. I'll disagree a little and say you are a superwoman, but no. I do agree that everyone can access peptides if they so choose to, and that peptides do open up so many opportunities.
Yes, exactly. So many opportunities, but always not thinking that it's an easy task. We have to study. Mm-hmm. And we have to study also the old literature coming from the Hubenweil, the first edition, the second edition, because everything can be revisited with the new technologies, improving the possibility to obtain good products, good peptides.
We are indeed sitting on the shoulder of giants. Nevertheless, we should never forget those who came before us, and we built a wonderful science.
Absolutely. I agree. It's so important to go back and look at original references. Do not just look at the references for the last 3 years.
Yes.
Find the original citation.
I started with Professor Luis Moroder, and I continuously speak with him by phone, and I have always something to learn. Another important person I am always in touch is Professor Michael Korev. So Professor Claudio Tognolo. These are people that are mentors, from whom we continue learning something, even if we are no more young. Yes.
Oh, I agree. Oh, thank you both so much for your insights. I really appreciate it.
Thank you.
Thank you, Wendy. Thank you very much for the opportunity, and hope to meet you soon.
Thank you for tuning in to this episode of Exploration Science. If you enjoyed this content, please like and share with your communities. As always, we appreciate your feedback as well as your suggestions for topics that you would like to see covered. Thanks again for tuning in.